So basically, it consists of this sphingoid base that's shown in red, and then a fatty acid acyl chain that varies from 14 to 26 carbons. And then it has a hydrophilic head group for more complex sphingolipids. For example, gangliosides like GD2 have this basic ceramide structure and then a sugar moiety attached to it. And ceramide is generated in response to many factors, including chemotherapy, heat stress, and TNF-alpha. We know it is elevated levels of ceramide are associated with apoptosis, differentiation, and actually when you add ceramide to neuroblastoma cells, you can actually induce differentiation. So this is just sort of showing how complex the pathway is. And my lab really focuses on the de novo pathway of ceramide generation, but ceramide can be made de novo. It can be made from the breakdown of sphingomyelin. Complex glycosphingolipids are made through glucosyl-ceramide synthase, and there is some data showing that cells that are multi-drug resistant have increased expression of glucosyl-ceramide synthase. Sphingosine 1-phosphate ceramide can be broken down to sphingosine, and when sphingosine is phosphorylated, sphingosine 1-phosphate sort of be the anti-ceramide, where ceramide is involved in apoptosis. S1P is involved in cell proliferation and also in angiogenesis. I'm going to touch on the ceramide synthases because some of the work we're doing in the lab is going to focus on that. So basically, the family of ceramide synthases will acylate dihydrosphingosine to form dihydroceramide. And in mammalian cells, each ceramide synthase has a specificity for a specific fatty acid. So eventually you get dihydroceramide with a different carbon group, and then through the action of the hydroceramide desaturase, a double bond is added and ceramide is generated. But I'll touch more on that a little bit later on the talk. Ceramide has many downstream targets, including NMYC and CMYC, and it's thought to act in many ways through activation of ceramide-activated protein phosphatases, particularly protein phosphatase 1 and protein phosphatase 2A. Dr. Thiel spoke about retinoids, and one thing we were interested, sort of got me interested in sphingolipids, was retinoids. We know the work from Dr. Mathey and Dr. Reynolds showing that the addition of retinoic acid improves survival for high-risk patients, and now treatment with retinoic acid, cis-RA, after transplant has become the standard of care. We know that retinoids have many downstream effects, including downregulation of NMYC, dephosphorylation of RB, and P27, in addition to other things. So a couple years ago, we looked at the role of ceramide in ATRA-mediated differentiation. And we treated cells with myriocin, which is a compound that actually blocks de novo ceramide generation. And you can see with ATRA cells are differentiated. When we treat with myriocin, we're able to block to some degree the amount of differentiation. And when we measured ceramide levels, we saw a marked increase in the very long chain ceramides, which are the C24, C24-1, which was blocked by myriocin. At the time, we were also looking at telomerase activity, and so that ATRA decreased telomerase in a dose and time-dependent manner, and this inhibition of telomerase was actually when you block ceramide generation by myriocin, we were able to block to some degree the effect, the decrease in telomerase activity. If you treated neuroblastoma cells with ceramide, you also saw that decrease. So that implicated ceramides to some degree their involvement in the differentiation of ATRA-mediated in neuroblastoma. We then sort of started working with fenretinide, and fenretinide is a novel retinoid that is planned in COG studies to be used as maintenance therapy in future high-risk studies. Mechanism not fully understood. Dr. Reynolds' group had reported that it activated de novo ceramide generation through serine palmitoyl transferase and ceramide synthase, whereas Penny Lavotte's group showed increases in acid sphingomyelinase. So we decided to look at fenretinide a couple of years ago, and what we found was actually that ceramide levels were, in a dose-dependent manner, ceramide levels decreased to some degree, but it was really the dihydroceramides that were being elevated. And we measured this through mass spectroscopy, and so it was across really, ceramide levels decreased to some degree, and all the dihydroceramides were increased. So that really, it was not species specific, but it was across the board. With our biochemists, we were able to make a sort of in situ assay to look at desaturase activity, where we had a dihydroceramide that was linked to a bromide salt, which we were able then to follow the conversion through LCMS. So we were able to look at the conversion from the C12 dihydropyridinium salt to the ceramide. And what we found here in this block here was that, and this was looking at 2 hours and at 6 hours, that we saw a dose-dependent decrease in the generation of the ceramide particle, suggesting that the effect was due to inhibition of the desaturase enzyme. We looked at the level of our mRNA and protein for desaturase, and it was unchanged, suggesting that either this effect was due to post-translational modification or due to a direct inhibition of the enzyme. We also looked at ATRA, and ATRA did not have any effect at all on desaturase activity. We inhibited the enzyme with siRNA and saw similar results to what we saw with fenretinide, where we saw an increase in dihydroceramides, just like we did with fenretinide. And then when we looked at inhibition of desaturase, either through siRNA or through low-dose fenretinide, if you treat high dose, you're going to get significant apoptosis and cell necrosis. What we saw cell cycle arrest at G1, and which correlated with dephosphorylation of RB. And when we treated with phosphatase inhibitors, specifically ocadaic acid, which blocked PP1, we felt it was related to protein phosphatase 1. So what was sort of exciting for us is that we found sort of a novel action of fenretinide that was related to sphingolipids. Since then, we've looked at sphingolipid metabolites, and Dr. Bob Curley at Ohio State University provided us these metabolites. The most abundant metabolite in plasma is 4-MPR. And this has been found to be inactive in cells. You have high levels after treatment of 4-HPR, but it's found to be an inactive compound. Dr. Formelli in Italy found the 4-oxo metabolite in patients with ovarian cancer, and found that this metabolite actually is a lot more active than 4-HPR. Dr. Curley provided us with a putative metabolite of 4-oxo 4-MPR, which was found to not have any effects on apoptosis or cell growth. And what we found as expected was that the two 4-HPR and this metabolite, which are both biologically active, they both decreased the enzyme activity, at least in situ. And whereas the inactive metabolite 4-HPR and the oxo 4-HPR had really no effect on the in situ enzyme activity in cells. And then when we looked at dihydroceramide levels, there was a dose-dependent increase with both 4-HPR and 4-oxo HPR, but not so in the inactive metabolites, which really suggested that the inhibition of the desaturase enzyme is really important to the action of fenretinide. We wanted to really determine whether it was really due to the enzyme activity, so we actually had a labeled dihydroceramide compound. It was labeled at the 4-5 double bond, which is this is what makes dihydroceramide, and the difference between dihydroceramide and ceramide is this double bond here. And you get formation of tritiated water. So we're able to have an assay in which we use Brett liver microsomes to really study the enzyme activity and see whether the finerenone is really inhibiting the enzyme or not in vitro. And this is sort of their methodology showing where we have both cold and hot substrate, and we're having an indirect method where we're able to measure the amount of labeled water to actually measure enzyme activity. So these are our data show with both the 4-HPR and the 4-oxo-4-HPR in our enzyme assay using rat liver microsomes, and we're able to see a dose-dependent inhibition in enzyme activity in the two compounds, and really very little effect with the other metabolites. And through more enzyme assays, we're able to show that both compounds were actually competitive inhibitors of the desaturase.